Bismillahirrahmanirrahim. My thesis entitled Phytochemical and Biological Study of some Vishya Faba cultivars, presented by Sohaila Saeed, demonstrator at Pharmacognosy Department, Faculty of Pharmacy, Alexandria University. My supervisory committee consists of Professor Dr. Masouda Al-Sayed Amir, Emeritus Professor of Pharmacognosy, Faculty of Pharmacy, Alexandria University. Professor Dr. Hala Helmi Zadout, Professor of Pharmacognosy, Faculty of Pharmacy, Alexandria University. Professor Dr. Samah Mustafa Al-Sahafi, Professor of Pharmacognosy, Faculty of Pharmacy, Alexandria University. And Associate Professor Dr. Hind Muhammad Dawood, Associate Professor of Pharmacognosy, Faculty of Pharmacy, Alexandria University. Introduction. Our target plant is Vishya faba, faba beans, or broad beans. Faba beans are considered as one of the richest sources of protein, dietary fiber, micronutrients, and bioactive phytochemicals. Faba beans are green legumes of family Fabaceae. It's one of the most important winter crop in Egypt. Faba beans are characterized by low glycemic indexes and fat contents, in addition to their health-promoting properties, including anti-cancer, anti-diabetic, anti-obesity, and cardioprotective effects. Faba beans contain vitamins, minerals, carbohydrates, proteins, amino acids, fat, dietary fibers, saponins, trypsin inhibitors, and other compounds. One of the most important components of faba bean is amino acids. The amino acids profile include two important amino acids, tyrosine and phenylalanine, which play an important role in management of Parkinson's disease. Faba beans contain bioactive compounds which can control blood glucose levels by inhibiting alpha amylase and alpha glucosidase. Polyphenols, as tannins, are also known to protect the human organism from chronic diseases such as cardiovascular conditions, diabetes, asthma, cancer, and inflammation. Main biological activities of Vishya faba: antioxidant, antifungal, antihypertensive, antileukemic, and anti-inflammatory. And there are two important biological activity which will we talk about in our study: anti-Parkinson and anti-diabetic activities. Diabetes. Diabetes is a chronic metabolic disease characterized by high blood sugar resulting from the body's inability to produce enough insulin or use it effectively. It causes severe long-term complications. Key types include type 1, type 2, and gestational. Diabetes can be manageable via diet, exercise, and medication. Faba beans are excellent, low glycemic, high fiber, and protein-rich food for managing type 2 diabetes. Faba bean contain bioactive compounds that can control blood glucose levels by inhibiting alpha amylase and alpha glucosidase, increasing beta cell regeneration, improve insulin release and resistance. They help stabilize blood sugar levels, improve insulin sensitivity. They also aid in reducing cholesterol levels and provide antioxidant benefits. Parkinson's disease. Parkinson's is a progressive neurodegenerative disorder caused by the degeneration of dopaminergic neurons in the substantia nigra of the midbrain. The pathogenesis of Parkinson's disease involves mainly a decrease of melanin pigment in the substantia nigra, accompanied by decrease of a biogenic amines, and the depletion of dopaminergic neurons in the substantia nigra and the corpus striatum. Symptoms of Parkinson's disease include tremor, speech changes, impaired balance, loss of smell, flatulence, and slowness of movement. Levodopa is still the most effective treatment for the disease. It's readily carried across blood-brain barrier, where it's taken up by dopamine neurons and converted to dopamine by the action of dopa decarboxylase. Levodopa is a naturally occurring amino acid mainly found in the embryo axis of faba beans, so the supplementations with such beans can be used for management of Parkinson's disease. A study in 6 adults with Parkinson's disease showed that consuming 1 to 200 grams, about 1 to 2 cups of faba beans, with the anti-Parkinson's medication carbidopa improved the symptoms as well as traditional drug. Our aim of the work: preparation of hydroalcoholic extracts from different plant parts of some Vicia faba cultivars. The next step: in vitro biological screening of the different extracts for their anti-diabetic and anti-Parkinson's activity if possible. Then, biologically guided fractionation for the most active fractions by in vitro biological screening on the resulted fractions. We will going to apply UPLC mass technique to the resulted fractions, then conduction of chemometric analysis to pinpoint the bioactive metabolites. Applying molecular docking studies on the bioactive metabolites to afford deep understanding to their interactions with the selected therapeutic targets. Then, isolation and structural elucidations of the selected bioactive metabolites, in addition to in vivo biological testing to verify their activities whenever possible. Our study design and research methods. The first step is sample collection and preparation. Vicia faba cultivars samples were collected from Nubaria Agricultural Research Station, and the collection process was established according to the national guidelines. The samples were then separated into organs, seeds, legumes, leaves, and stems. Maceration of Vicia faba selected organs in 70% ethanol and in hot water in an ultrasonic bath apparatus to gather the majority of constituents. After obtaining the hydroalcoholic extracts, they will be concentrated till dryness under reduced pressure using a rotary evaporator, while aqueous extract will be concentrated via lyophilizer. The next step: mass spectrometry based metabolomics. The next step involves chemical metabolomic profiling of the extracts of different candidates using different chromatographic and spectroscopic techniques, and subsequently establishing appropriate statistical models for candidates discrimination. Our present study will employ UPLC mass metabolites fingerprinting technique for the first systematic broad-scale metabolites characterization of faba bean sample. Multivariate statistical analysis, such as principal component analysis, followed by orthogonal projection to latent structure discriminant analysis, are extensively utilized for LC mass data analysis to verify trends and effectively illuminate compositional heterogeneity among Vicia faba samples. Then, biological activity investigation. Different faba bean samples of different cultivars will be screened for anti-diabetic activity using alpha amylase, alpha glucosidase enzyme. Anti-Parkinson-induced rat models will be established for examination of the effect of faba bean extracts in the disease improvement. Scientific and technological innovation. Our research aims to expand the knowledge about LC mass-based metabolomics approach integrated with multivariant statistical analysis that will reveal the compositional heterogeneity in metabolite profiles of faba bean extracts and pinpoint the most relevant chemical biomarkers, which in turn signify their role in the management of diabetes and Parkinson's disease that affect millions of people worldwide. Isolated biomarkers will serve as cornerstones and starting points for the development of successful anti-diabetic and anti-Parkinson agents as drug candidates in the future. Expected outcomes. Our research aims to sparkle an interest toward the exploitation of faba bean extracts, as they are an extraordinary reservoir of different classes of secondary metabolites with the different pharmacological activities. One of them is the anti-diabetic potential. Worth mentioning, constituents isolated from natural origin may be successfully lead compounds that can be modified to improve their pharmacokinetic behavior and produce anti-diabetic and anti-Parkinson drugs in the future with minimal side effects. These potentially active natural products could be incorporated into preclinical development studies to assure its safety as therapeutic agents. Eventually, there will be more and more awareness of suitable use of these natural resources rather than synthetic and processed products, with the harmful side effects to the consumer in the coming years. Thank you.